Human aging is characterized by a state of chronic inflammation, termed inflammaging, for which the causes are incompletely understood. It is known, however, that macrophages play a driving role in the establishment of this condition by promoting pro-inflammatory rather than anti-inflammatory responses. Overall, both environmental and genetic factors have been implicated with this condition, with the most important contributors being the genes of interleukin 6, IL-1 receptor antagonist, and TNF-alpha. It remains unclear, however, if genes involved in the signaling cascades of those cytokines could also be involved with inflammaging. Our group focuses on the study of a serine threonine kinase termed TAOK3. This gene was associated with inflammatory and autoimmune conditions such as rheumatoid arthritis and lupus. Yet, the functional role of TAOK3 in inflammation has remained unexplored. We thus used a TAOK3 deficient mouse model to investigate the potential importance of this gene in chronic and age-related inflammation. We found that a higher proportion of knockout animals compared to wild type develop severe skin ulceration with age. Furthermore, these aged mice also had a severe increase in their spleen size. Histologic analyses later revealed that the increase in the spleen was due to an accumulation of the white pulp zone, a zone that is predominantly populated by immune cells. It remained unknown, however, which cell type differentially composed these spleens. Flow cytometric analyses revealed that both young and aged knockout animal had more myeloid cells and less B cells in their spleens. To understand whether or not the accumulation of myeloid cells was the result of a difference in earlier stages of hematopoiesis, we took a closer look at the progenitor cells found in the bone marrow. These observations revealed a drastic shift toward myeloid-committed progenitor cells, or MPP3, rather than lymphoid-committed progenitor cells, MPP4, in the bone marrow of aged TAOK3 deficient mice. Peripheral blood specimens also had a similar increase of myeloid cells, as was previously shown in the spleen. Overall, these data suggest that TAOK3 deletion causes a shift in the type of immune cell produced in the bone marrow, favoring myeloid cell production and their subsequent peripheral accumulation. Higher levels of pro-inflammatory factors such as IL-6, TNF-alpha, and CXCL9 were also detected in TAOK3 knockout mice using cytokine multiplexing. Furthermore, we uncovered that the increase of those markers was also more pronounced in female animals, not unlike what is seen in humans in diseases that have strong sexual dimorphisms, such as lupus. These data support initial hypotheses that increased age-related inflammation could be observed in TAOK3 deficient mice. We next sought to evaluate the functional relevance of TAOK3 deletion in macrophage functions, given their central role in inflammaging. We found that TAOK3 deficient bone marrow-derived macrophages, or BMDMs, has stronger pro-inflammatory responses upon treatment with LPS and interferon gamma. Indeed, polarization led to increased IL-6, INOS, and also TNF-alpha production in knockout BMDMs. Two outstanding questions remain, however. One, whether the kinase activity of the enzyme was responsible for the phenotypes at hand, and two, whether the deletion of the kinase was more important during the differentiation or the polarization phase of BMDM cultures. To answer those questions, we use a TAOK-specific inhibitor called compound 43. We first observed that the treatment of BMDM cultures with increasing concentrations of compound 43 led to increased frequency of F480 positive macrophages in those cultures. Next, we designed our assays in a manner that the inhibitor was either put during the differentiation phase or the polarization phase, or both. As expected, we found that inhibition led to increased iNOS production in all groups. What was unexpected, however, was that treatment with compound 43 during the differentiation phase had a stronger impact on the mounting of pro-inflammatory responses compared to treatment during the polarization phase alone. Indeed, TNF-alpha was only found to be increased in groups that were treated during the differentiation phase. These observations mean that TAOK3 plays the majority of its function in limiting the setup of pro-inflammatory programs during the differentiation of macrophage. Essentially, TAOK3 deficiency promotes the accumulation of monocytes in the periphery, their conversion to macrophages, and their subsequent secretion of pro-inflammatory factors. These findings illustrate the role of TAOK3 in age-related inflammation and highlight the importance of genetic risk factors for this condition. I'd like to end it here by acknowledging all the lab members involved in this project, my supervisor Dr. Michel Tremblay, and all the supporting agencies. Thank you for your time and enjoy the rest of the meeting.